All right, next section on the T cell receptor complex. So, here are some of our vocabulary terms. We've got Luck and Fin. So, these are both Sarc family tyrosine kinases. So, these are things that are going to make phosphotyrosine on certain proteins. The Luck protein is going to be attached to the tail of a CD4 or CD8, and so that's going to phosphorylate some of those ITAM motifs. And yeah, we're going to have to see what Finn phosphorylates. I don't remember. I just remember hearing about it. Okay. We're also going to have run into C-terminal Sarc kinase, which is CSK. And yeah, it is going to phosphorylate the Sarc family tyrosine kinases. So it's going to be an activator of some of these kinases like Luck. Luck or lick, I guess you could say it either way. However you want to pronounce a word without a vowel. Okay, and then tyrosine phosphatases are going to be enzymes that remove the phosphate groups. So let's meet some of these. All right, CD45 is going to be one of these tyrosine phosphatases. So CD45 is going to remove a phosphate from Luck. And by removing that phosphate, we're then going to activate Luck because the phosphate is bound and it kind of locks the Luck protein down. And so this is the first step to activating Luck, which then activates the T cell receptor, which then can bind to all these other proteins like ZAP70 and can set off further activation steps. So here we go. Here's the first part of this. So this is in the top panel over here at the left. This is inactivated Luck. And so you can see all these parts here, the two cysteines, the SH3, the SH2, the little squiggly bit, the kinase domain, and the little tail that has the phosphate on it bound to the SH2. All those are part of the Luck protein. So this is not anything else. These are just all the pieces of Luck. We're kind of zooming in on the Luck protein now to see how it is activated. At the top, you can see there is a zinc atom, and it's going to be coordinated, hydrogen bonded essentially, between 4 cysteine atoms. And these are rather cysteine amino acids, pardon me. And so this is going to be a thing called a zinc finger. Usually the 4 cysteines, or, it can be a mix of cysteine and histidine residues. Usually these things would occur on the same chain and just kind of wrap around the zinc. In this case, it is actually a zinc finger formed from 2 different protein chains. Kind of neat. And so the first 2 cysteines will be either on the CD4 or CD8 protein. They would both look the same in this part. They have that same double cysteine motif. And the other 2 are going to be on Luck. And together, those are going to hold on to a zinc atom, which is going to serve as the bridge between CD4 and CD8. And so this may be part of the reason behind why people tell you you're supposed to take zinc to prevent colds and stuff. The truth is most of that doesn't get absorbed, and you already have enough zinc floating around in this and so many other proteins that it really doesn't make much of a difference whether you're taking dietary supplemental zinc. But theoretically, if you completely zinc-starved a person, this would be one of the thousands of things that could go wrong with them. All right. So now, here we've got 2 possible ways. We've got an activation step, which is CD45, and we've got an inactivation step, which is CSK. Let's see. So CD45 is going to be the activating protein, and CD45 is a phosphatase. And so the phosphatase is going to remove that little phosphate on the tail of the kinase domain, and that is going to prevent the SH2 from binding. So the SH2 is bound to this phosphate, and that kind of twists and locks up the kinase. Imagine the kinase kind of has its arms pinned behind it, something like that. That's how it is when the phosphate is there, and then when you release the phosphate, then boom, it can spring out, and the rest of it can be flexible and move where it needs to and do the kinase activity that it needs to do. C-terminal Sarc kinase, which is CSK, is going to be the kinase that puts the phosphate onto Luck and shuts it down. So high CSK is going to inactivate luck, high CD45 is going to reactivate luck, and it's kind of going to be a battle between these two for whether luck is available to do phosphorylation or not. Once luck is primed, that is, that it has its phosphate removed, it will now activate some of these ITAM motifs by putting a phosphate onto the tyrosine to make phosphotyrosine there on those zeta chains, and also on all 4 chains of CD3, which also have those ITAM motifs. Okay, next protein, ZAP70, which is the zeta chain associated protein, and it's about 70 kilodaltons, I think. That's why it's ZAP70. This is going to be a tyrosine kinase. So again, it's going to put phosphates onto tyrosines. It's going to be found in T cells, but it's not going to work unless it is bound to a doubly phosphorylated zeta chain of a T cell receptor. So that's, this is going to bind to the ITAM, the tyrosine motifs, when they are phosphorylated. And once it binds to those, then it will be activated and it will start to put phosphates onto other things. And so this is going to be the next step. The previous step was we're going to activate CD45, which is going to turn on our luck, or we're going to activate CSK if we need to turn off our luck, basically. And then once luck is turned on and it puts those phosphates onto the zeta chain, ITAMs, then ZAP70 can bind to those, and then we're off. So, if you were curious, this is what a ZAP70 looks like. It has 2 SH2 domains, the sort of bluish one here, and the sort of greenish one that you can see up here. And then the rest of this thing is going to be the kinase. Shown here, the 2 yellow dots, which are here and here. Those are going to be the tyrosines that can get phosphorylated. All right. So, next little vocabulary word is going to be called the FC gamma receptor number 3. You're like, okay, that's another weird word. But you can break this down. You know what this is. So FC, you remember that? That's part of an antibody, right? It's going to be the constant region of the antibody, which means like the handle of the antibody, not the, prongs that stick out like a Y, but the bottom of the Y. And FC gamma would be binding to the FC, the constant region of IgG. And then this is just receptor 3 for that, and its other name is CD16. I like FC gamma receptor 3 better as a name because it kind of explains what it is. If you can see the name there, you can unpick the thing, unpack it, and you've got the whole idea. So this is going to be one of those proteins that binds to IgG molecules when they are aggregated together. And so we will show what that looks like in a slide or two. Okay, so there are a variety of receptors that also associate with ITAM-containing chains. So ITAMs are not just on CD3 and the zeta chain attached to the T cell receptor. They're also on many others. So for example, FC gamma receptor 3, the one we just mentioned, has these little either gamma chains or zeta chains that will attach to it and will carry ITAM motifs. You find that on NK cells, macrophages, and neutrophils. NK cells will have another thing called an NKG2C, which is an NK cell glycoprotein, type 2C, D, and E, whatever, which is also known as CD94, and so that will also associate with another thing. It's going to be linked together by a disulfide bond. It's going to have ITAM motifs. It's going to be a lot like a zeta chain, but it's a different zeta chain. And here we go, FC epsilon receptor 1. Now that's a neat one. Can you unpack that yourself? Let's wait a sec. So FC, constant region of an antibody, right? Yeah, of course. Epsilon, constant region of an IgE antibody. We haven't talked much about those except to say that they are heavily involved in allergy. And the process of allergy, I think my nose is a little stuffed up at the moment, is going to be driven by mast cells and basophils for example. And these mast cells and basophils are driven, they help drive, rather, the allergy process by binding to the tail of an IgE that is stuck to something. And so if the IgE binds to pollen, then your mast cells are going to bind to IgE. They're going to think pollen is a bad guy, and they are going to make a response. And that's what you will perceive as allergy, because there's really not that much left in the world that IgE needs to bind to. IgG pretty much takes care of the blood. We got all those 4 types. IgA is going to be on the mucosal surfaces and a little bit in the bloodstream. And IgM is the early starter antibody that looks like a Chinese throwing star. No, ninja throwing star. And that's also going to be found in the blood. So between those things, they actually do a very good job of patrolling your body. And IgE is kind of an evolutionary relic. But the Fc epsilon receptor 1 on your mast cells and basophils, that's the reason why we have allergies. There we go. Okay, so next one, LAT. Remember, I think of this one as being long and tall, but its actual name is linker for the activation of T cells. This is just a linker. It's a big, long protein that has several tyrosine residues that can be phosphorylated by ZAP70, and it will serve as a platform. Other proteins will bind to it, and so it can hold all these other proteins together. It's kind of like a frame that you can clip other proteins onto to make this sort of customized, very temporary immunological machine. And this happens as part of an immune response. So ZAP70 is going to phosphorylate LAT, and there'll be other proteins that dephosphorylate LAT, or that have phosphatase activity. SLP76, SLP76 is going to be another sort of scaffold protein. And it's also going to be involved in some of these pathways. There are a lot of these proteins whose job is to get phosphorylated and then serve as a scaffold of some sort for other proteins to bind to. This sort of thing goes on all the time in cell biology. But let's just meet a couple of these guys. Okay, so ZAP70, which is shown right here, bound to the zeta chains. It's the zeta-associated protein, remember? And it's a kinase. You can tell from the way they've drawn it and from just remembering that it's a kinase. So that is going to phosphorylate, add phosphates to the tyrosines on the LAT protein. And it's also going to phosphorylate SLP76. And so that's going to allow us to form a couple of different components. So this ZAP70 activation is going to eventually lead to AKT activation, which is going to make PIP3, and there we go. So AKT is going to be activated because we have PIP3 here, because we've expressed the PI3 kinase, which turns PI2 or PIP2 into PIP3. We are going to activate PLC gamma, which you remember is going to cleave our little PIP3. It's going to cut the head off from the tail and leave us with a diacylglycerol and a quadruple phosphorylated phosphatidylinositol, or I guess it's just 3 extra phosphates besides the one that normally is on phosphatidylinositol. And you can see that's going to be bound to SLP76. There we go. It's going to interact. SLP76 is going to bind to GADS down here, and GADS is going to bind to LAT. And so GADS is holding on to SLP76, which is holding on to PLC gamma. PLC gamma is also holding on to LAT on this side. And so, yeah, together, LAT and SLP76 make a nice little cradle where PLC gamma can do its job. Now, we've got separate slides on each of these pathways coming up, but this is like a little introduction to those pathways, I would say. Okay. Then we've also got VAV over here. VAV is going to be another one of these proteins that's going to bind to PIP3, so our little signaling molecule, and it's also going to hold on to SLP76 once it is phosphorylated. And VAV will then go and signal for actin molecules, which are part of the cytoskeleton, to start to polymerize. We'll start to make more actin molecules. This is going to have the effect of causing the cell to change shape and to move. So take something like a platelet cell. It just looks like a little tiny, kind of like a dinner roll when it's just floating around in the bloodstream. But when it's activated, this goes really spiky. And it's because of an activation process like this, because the actin starts to get polymerized, and it makes little pointy chains. This is also the way that cells move. If a cell wants to move, it will actually dissolve its skeleton at kind of the back end of the cell and then make new skeleton, which pokes out in the direction that it wants to move. And that's basically how they move, by dissolving and reforming their skeleton. So that's kind of a neat thing. And yeah, valve activation is going to lead to that. And then over here we have ADAP, and we're going to be able to recruit that because it's also got a phosphotyrosine on it, and it's going to bind to SLP76, which is bound to GADS. GADS is just like a little linker, I would say, for these two platforms. Yeah, that's going to start a different pathway, which is going to lead to integrin sticking and clustering together. So integrin is a protein that you're going to find on the outside of some white blood cells in particular, and this allows the white blood cells to stick to other cells and actually even squeeze in between the other cells sometimes. So as white blood cells are patrolling in the body, they are circulating around with your blood. Occasionally there will be a signal and they will stop, they'll drop out of the bloodstream and grab onto the side using their integrins, and then they can roll along the side looking for the source of the problem, and when they find it, they will slip in between some other cells and go off and yeah, attempt to deal with the infection or whatever the problem is. So yeah, and this is going to be driven by integrin, which is going to be itself be driven by ADAP. All right, and believe me, it takes a lot longer to explain all these parts and what's going on than it does for it to happen. This whole process can happen in a matter of seconds, yeah. But yeah, it takes a little longer to explain it. Okay, so phospholipase C gamma, or PLC gamma, we remember this one, right? Yeah. It's going to be a key enzyme in signaling, and it's going to cut off the phosphatidylinositol triphosphate from the diacylglycerol tail. And the diacylglycerol, or DAG, DAG, is going to be it's a lipid that also serves as a signaling molecule. It looks like a membrane phospholipid molecule, but it's missing its head. So it's like a little headless horseman galloping around. And yeah, if you were to see one of those, I think you'd pay attention as well. The immune system certainly does. Okay, inositol 1,4,5 triphosphate, which is IP3. This is going to be the head group of PIP3. So this is just the phosphatidylinositol head group with 3 extra phosphates on it after it gets cleaved off by PLC gamma. And it is going to float through the cytoplasm, and it's going to bind to the endoplasmic reticulum membrane, and there it is going to cause the release of stored calcium in the cytosol. So, okay. That's kind of the next little step. And again, we're going to have pictures that show all this stuff in just a minute. But yeah, for now, let's just meet the players. Okay, STIM1 is going to be a calcium sensor in the endoplasmic reticulum. And whenever we start to run low on calcium in the endoplasmic reticulum, it's going to open up the plasma membrane calcium channels, basically. And calcium release activated calcium channels are going to open up and let calcium go into the cell while it's responding to antigens. So this is a way that the cell is going to recharge its calcium stores. Protein kinase C theta, that's what that O with a line through it is a serine or threonine kinase, and it can work on either one. It's going to be activated by diacylglycerol. Now, do you remember diacylglycerol? It's the little lipid tails, right, of our PIP3 molecule. So after we cut it in half, one half, the diacylglycerol is going to go and activate protein kinase C gamma subunit. All right, and then SCID is severe combined immunodeficiency. This is a type of immune deficiency where you don't have either B cells or T cells. It's generally going to be fatal. This is going to lead to, it's going to be one of those conditions where a person could survive, but they're going to have to be in some kind of like filtered plastic bubble for their entire lives. You can get and make skid mice. Yeah, these things have no B cell or T cell response. So they're missing proteins that are involved in both B cell and T cell signaling. I believe there are different ways to actually make these things, yeah, they are useful in science because they can show you what the other components of the immune response are doing, like what their contributions are. They're sort of like, well, what if we take out B cells and T cells, so they're not part of the equation? What else? What do the rest of the cells do? Because otherwise, B cells and T cells tend to dominate an immune response. Okay, so rewind back to where we were. All right, so you remember that our T cell receptor, and here it is bound to a MHC either class 1 or 2, depending on which one we've got. And so there'll be either a CD4 or CD8 over here, depending on whether we're bound to class 2 or class 1. We've got our CD3 complex here with the little ITAMs on it. Great. We've got Luck just off screen. You just can't quite see it, but it would be right over here. And it would have phosphorylated the ITAMs here on the bottom of CD3 and the zeta chain and the other CD3 chains. Then we've got ZAP70, which is now able to bind to the zeta chain now that it is multiply phosphorylated. ZAP70 is going to put the phosphates onto SLP76 and onto LAT. Now, we've got our GADS protein that can only bind to a phosphotyrosine because it has an SH2 motif, and that's exactly what they do. And so it's going to bind to LAT, which is the long and tall protein right here, and to SLP76, which is going to be hanging off the side like a curvy little banana. And then PIP3 is going to be floating around in the membrane at the same time. So this is going to be made by a separate process involving the activation of the PI3 kinase. And once you've assembled SLP76, LAT, and GADS, that is actually going to be something that promotes activation of PI3 kinase, which will make our PIP3, of course. Okay, so once we have some PIP3 in the neighborhood that we've made, because we made this particular complex, now we can recruit ITK, and we can also recruit PLC gamma. And PLC gamma is going to do its job, but only after ITK activates it. So ITK is going to be a kinase, and it is going to activate PLC gamma. PLC gamma would love to cut the head off this phosphatidylinositol triphosphate, but it can't do it until it is activated. So yeah, you assemble the complex with GADS in the middle. You bind the ITK on the side of the complex to the phosphotyrosine on SLP76. Then the ITK phosphorylates PLC gamma, which is also bound to a PIP3 molecule. And then once it is bound, it's going to cut this PIP3 molecule into two parts, which are going to be the phosphatidylinositol triphosphate, or IP3, and the other part's going to be called diacylglycerol, which is just the tails. And I think that's going to be on the next slide. Yeah, some of that. There we go. Yeah, it is. Okay, so it's over here at the side. It's a little small, but yeah, we can cleave either PIP2 or PIP3, yeah, into diacylglycerol and either IP3 or IP2. And that's going to go down, and it's going to bind to an ion channel on the ER. And it's going to cause the calcium in the ER to leak out into the cytoplasm. And so we're going to deplete the calcium that would normally be stored up in the ER. The ER normally works very hard to store up some calcium. And so then just this is what happens after. So our we're going to have several of these molecules like protein kinase C theta chain and RAS GRP. They are going to bind to the diacylglycerol, and they will then become activated, and they will then go out and do other things. They're serine threonine kinases. Eventually they are going to activate NF-kappa B. So that's a process that we covered earlier, but this is another way that we can get to NF-kappa B activation. By, of course, activating IKK, which then activates I-kappa B by phosphorylating it, which causes I-kappa B to be shredded, fall apart, which then frees up NF-kappa B to go into the nucleus and cause transcription. There we go. It's a lot, right? And then just so you can see how it cleans up this ORAI1 is going to be the protein that's going to open back up and let in more calcium back into the cell after we've depleted our calcium stores. And then the cell will continue to store away. It'll put this calcium from the cytoplasm back into the ER and eventually restore the balance as it was before we activated it. So why release calcium? Good question. Where does it go? Okay, let's talk about it. So NFAT is the nuclear factor of activated T cells, and it is a transcription factor, part of a family of them. Calmodulin, which, you may even remember, is a calcium binding protein, going to be activated by calcium binding, and it's going to stick to a bunch of different enzymes, and calcineurin is going to be another phosphatase. So it's going to take off the phosphates from serines and threonines, and it's going to have a role in T cell receptor signaling. So calcineurin is going to lead to stopping the T cell response. So this is like the other end. After it's over, calcineurin is going to come in, take away some of these phosphates, and shut all of this stuff down that we just learned about. Okay. Cyclosporin A is an immunosuppressive drug. It's sometimes given to transplant recipients, for example, and it's going to inhibit signaling from the T cell receptor. So it's going to block T cell receptor function, and by binding to a thing called cyclophilin A. And cyclophilin A is going to inhibit calcineurin. So if you're curious why your, parents, grandparents, whatever, are taking cyclosporin A, this is exactly why. And tacrolimus, neat, that's fairly modern, is another immunosuppressant drug. And it's going to form a complex with FK binding protein, and that's going to inactivate T cells by, once again, inhibiting calcineurin, the phosphatase. Okay, so the idea is that we've got our ER, I believe, at the top here. We've got a cytoplasm in gray down here. And so we start out with lots of calcium in the ER, not so much in the cytoplasm. So calcium binding proteins in the cytoplasm just have to kind of hang out, and wait. All right, next step. Once we send IP3 molecule, the sort of cut in half PIP3 molecule or PIP2 molecule, once we send that over to open up the calcium channel, it's going to open that up and it's going to dump calcium from the ER into the cytoplasm. Rather, it will allow the calcium to equilibrate either side of the membrane, which is exactly what it does. Now, calmodulin has been waiting here, but it wasn't able to assume its correct shape until it bound calcium. So once calmodulin binds, it then opens up and it becomes a good little platform where calcineurin can bind. Calcineurin will take all of the phosphates off of NFAT, and then once NFAT is fully dephosphorylated, it can then go into the nucleus, and boom, it's going to activate gene transcription. And so that's going to be part of the process. That's one process that can lead that's going to be involved in T cell activation. Okay, I think that's a good place to pause and maybe go back and rewatch some of this and get all of this into your mind. This is the complicated chapter, but it's got some really good stuff in there. And once you can practice this a few times, really get it into your mind, I think, I think this is going to click, and I think this is going to work for you. So thanks very much.